Let's deep dive into a mind-blowing YouTube podcast created by artificial intelligence, where you will hear conversations by two AI agents in a way you've never imagined before. This isn't just a podcast, it's a journey into the future, crafted by AI, designed for you. Ready to have your mind blown? Let's begin.
Welcome to a deep dive into the world of DNA. We're going to be exploring Horace Freeland Judson's The Eighth Day of Creation.
Oh yeah, The Eighth Day of Creation.
To uncover how scientists unlocked the secrets of life itself. Pretty amazing stuff.
Judson's book is so fascinating because it really delves into the human drama behind all these big scientific discoveries. It's not just facts and figures.
Right.
It's about the rivalries, the collaborations, those moments where everything clicked for these researchers. It's like reading a detective story.
Okay, so let's go back in time a bit. We're talking the 1950s. What was the scientific landscape like back then?
Well, you have this intense race going on to figure out the structure of DNA.
Which is...
like the instruction manual for life.
Right, the blueprint, the whole thing, yeah.
Exactly. And you had these key players like James Watson and Francis Crick at Cambridge.
Okay.
And Rosalind Franklin and Maurice Wilkins over at King's College.
Ah, interesting.
All trying to crack the code, so to speak.
Now, I remember learning about Rosalind Franklin's famous Photo 51, that X-ray image of DNA. That was a pretty big deal, right?
Absolutely. Photo 51 provided crucial evidence for a helical structure, though sadly, Franklin's contribution wasn't fully acknowledged at the time.
Oh, that's a shame.
It does raise some questions about how credit is given in scientific discoveries.
For sure. So they figure out DNA is this double helix.
Yes.
But what does that actually tell us? It's a cool shape.
Right.
But how does it work?
Well, it's so much more than just a shape. Think about it like a twisted ladder.
Okay.
And the rungs of this ladder are made of pairs of molecules called bases. A always pairs with T and G with C.
Okay.
Now, because those two strands are complementary, it explains how DNA can copy itself perfectly.
Ah, I see. So one strand acts like a template to make a new matching strand.
Exactly. It's like unzipping a zipper and each side becomes a new half.
Oh, that's a great way to think about it. So
Yeah.
that's how we go from one cell to trillions. Incredible.
It really is.
But okay, we've got the structure down, but what about the language of DNA? Like, how do those letters, those bases, actually get translated into, all the stuff that makes us, us?
Right. So that's where cracking the genetic code comes in.
Okay.
We're moving into the 1960s now.
All right.
And scientists are on this new quest to understand how the sequence of those bases in DNA actually tells the cell how to build proteins.
Okay, now proteins, those are the workhorses of the cell, right?
They are. They do so many things, building and repairing tissues, transporting molecules, fighting off infections.
Right. It's a long list.
Huge. So deciphering the genetic code was like unlocking the instruction manual for building and running an entire organism.
Wow, that's a big deal. So who were the key players in this endeavor? Cracking this code, I mean.
Well, it started with a brilliant experiment by Marshall Nirenberg and Heinrich Mathai. They were working with this synthetic RNA. It's like a simplified version of DNA.
So if DNA is like a hardback book, RNA is the paperback.
Precisely. It carries a similar message, but in a more accessible format. Anyway, they created a chain of RNA made of only one base, uracil, over and over and over again. And what they discovered was that this RNA chain produced a protein made entirely of one amino acid called phenylalanine.
So hold on. They figure out that UU 3 uracils in a row code for the amino acid phenylalanine.
Yes.
That's like cracking the first word in the language of life.
It was monumental. And it kicked off this race to decipher the rest of the code. And by 1966,
Okay.
Nirenberg and Hargub Khorana had mapped out the entire genetic code.
Wow.
It's like the Rosetta Stone for understanding how DNA builds life.
That's amazing. Didn't they also find that the genetic code had some, redundancy? Like nature was repeating itself a bit?
You're right. They found that multiple combinations of those bases, what we call codons, can code for the same amino acid.
Wait, so why would nature do that? Seems a little inefficient.
Well, actually, it's incredibly clever. It's like a built-in error correction system.
Oh, interesting.
If there's a small mistake in the DNA sequence, there's a good chance it won't even change the protein that's made.
Huh. So it's like a backup plan. Pretty cool. But okay, so we have DNA in the nucleus, the control center of the cell.
Right.
But proteins are made out in the cytoplasm. That's like the cell's workshop. So how does the information get from point A to point B?
That is where mRNA comes in.
mRNA.
Messenger RNA. Think of it like a courier, carrying the blueprints from the DNA out to the ribosomes.
And the ribosomes, those are the protein-making factories.
Exactly.
Okay. I'm picturing a tiny messenger sprinting across the cell with this little blueprint tucked under its arm. But how did scientists figure out this whole mRNA thing?
Judson describes a clever experiment by Sidney Brenner, Francois Jacob, and Matthew Messelson. They used radioactive labeling to track newly made RNA molecules.
Like putting a little tracker on them.
Exactly. And they were able to follow these labeled molecules as they move from the DNA to the ribosomes. It was proof that mRNA was carrying the genetic instructions.
So they could actually see the information flow happening. That's incredible.
It is.
So this whole process, DNA to RNA to protein,
Yes.
it's like the fundamental principle of life, right?
It is. We call it the central dogma of molecular biology.
The central dogma.
This flow of information is how every living thing functions, from bacteria to humans.
Okay, so we've gone from the basic structure of DNA to this intricate system of information flow. It's mind-blowing.
It is.
And we'll be right back after a quick break to delve even deeper into this incredible world of DNA.
Welcome back to our deep dive into the world of DNA. This whole idea of the central dogma, DNA to RNA to protein, it's like a universal language. Every living thing uses it.
It is fascinating. Makes you realize how much we have in common with even the simplest organisms.
Right. It's all about information flow. But here's a thought. Are all our genes, active all the time?
I never thought about that. Is the whole DNA instruction manual being read constantly?
That's a great question.
Yeah.
And it takes us to one of the most elegant examples of how genes are regulated, the lac operon.
Lac operon.
I know it sounds a bit technical, but trust me, it's a really cool story about how bacteria manage their energy.
Okay, bacteria and energy, I'm listening.
All right, so imagine E. coli bacteria in your gut. They can use lactose, the sugar in milk, as fuel.
Okay.
But making the enzymes to break down lactose takes energy, right? So they have this system where they only make those enzymes when lactose is actually present.
Oh, wow. So they don't waste energy making stuff they don't need.
Exactly. It's like a built-in efficiency mechanism.
Hmm.
And what scientists Francois Jacob and Jacques Monod figured out is that there's a little section of DNA, the lac operon, that controls this whole thing.
So it's like a switch for those genes.
Precisely. Normally, there's a repressor protein bound to the DNA, blocking the genes for lactose metabolism. But when lactose shows up,
Okay.
it binds to the repressor and basically kicks it off the DNA. Now those genes can be turned on, and the bacteria start producing those enzymes.
So lactose comes in, repressor gets booted, the party starts. That's a great analogy. But this gene regulation thing, it's not just a bacteria thing, is it? Our cells do it too, right?
Absolutely. This whole idea of genes being turned on or off in response to signals is fundamental to how all living things work. Think about how our bodies develop,
Right.
how we respond to stress, how our cells know what to do. It's all about gene regulation.
Makes sense. But hold on, we've been manipulating genes for a long time before we even knew what DNA was.
That's true.
I mean, think about dog breeding or those giant pumpkins people grow for competitions.
Exactly. Judson calls this artificial selection.
Artificial selection.
For thousands of years, farmers have been selecting and breeding plants and animals to enhance specific traits. It's like shaping evolution with our own hands.
Yeah, like if you keep planting seeds from the biggest tomatoes,
Right.
you eventually get even bigger tomatoes. But are we actually manipulating genes when we do that? I mean, it's not like we're in there splicing DNA.
Not directly, no.
Yeah.
But you are influencing which genes get passed down to the next generation. Think about Chihuahuas and Great Danes.
Oh, yeah, huge difference.
Totally different sizes, but both dogs. And it's all because of variations in their DNA that have been selectively bred over generations.
So it's like a slow and steady kind of genetic engineering.
Exactly. But things got a whole lot faster in the 1970s with recombinant DNA technology. Now we can actually cut and paste genes, combine DNA from different organisms.
Now that's some serious genetic engineering. What were some of the first things they did with that technology?
Well, one of the biggest early achievements was the production of human insulin in bacteria.
Wait, so they took the human gene for insulin and put it into bacteria?
Yes. They created little bacterial factories that could pump out human insulin.
That's incredible. And that made insulin much more accessible to people with diabetes. Right?
Exactly. It was a game changer for treatment. And this opened the door to so many possibilities, not just for medicine, but agriculture, even industrial applications.
I can imagine. Like genetically modified crops.
Right. Crops that are resistant to pests or herbicides.
Hmm.
That's already happening. And we've also seen gene therapy emerging. The idea of actually fixing faulty genes to cure diseases.
Now that sounds like science fiction.
It does, doesn't it?
But is it actually working?
Well, there have been some really exciting breakthroughs. Gene therapy is now being used to treat some forms of inherited blindness and severe combined immunodeficiency.
Wow, that's amazing. It feels like we're entering a whole new era of medicine. But where do we go from here? What are the next big challenges?
The potential is huge, but there are still obstacles. We need safer, more effective methods for delivering genes into the right cells. And we need to understand the long-term effects of gene therapy better.
So even with all this amazing progress,
Right.
there's still so much we don't know. I guess that's the nature of science, isn't it? Always pushing the boundaries.
It is. Judson's book really captures that spirit of exploration. And he leaves us with this sense of responsibility, too. If the 20th century was about figuring out the secrets of life,
Yeah,
the 21st century is about using that knowledge wisely.
that's a good point. So we've covered a lot of ground here, from the structure of DNA to manipulating genes, and now even the possibility of curing diseases. It's incredible how much our understanding has changed in just a few decades.
It really is. And I think it's important to remember that science is a human endeavor. It's driven by curiosity and collaboration.
And sometimes even a little bit of healthy competition, right?
Exactly.
Those stories about the race to discover the structure of DNA, that was intense.
It was. But it also highlights how these breakthroughs can lead to amazing advancements that benefit all of humanity.
Absolutely. It's a reminder that we're all connected by this shared language of DNA. It's the blueprint for life itself.
And we're only beginning to understand its full potential. It's pretty amazing stuff. Think about being able to replace like a broken part in a machine.
Yeah, like a tiny cellular mechanic. So are there diseases that have actually been cured with gene therapy?
Yeah, there have been some remarkable successes, like certain types of inherited blindness and severe combined immunodeficiency. You might have heard of bubble boy disease.
Yeah.
That's one that gene therapy is being used to treat now.
But it really feels like we're on the cusp of a whole new era of medicine. Where do we go from here, though? What's next for gene therapy?
The potential is huge.
Yeah.
But there are definitely challenges too, like figuring out how to deliver genes safely and effectively into the right cells.
Right, right.
And we need to understand the long-term effects better. It's still early days.
Yeah, Judson's book really captures that sense of, like, ongoing exploration, doesn't it?
It does. He leaves us with a sense of wonder, but also a call to action. He says if the 20th century was about unraveling the secrets of life, the 21st century is about using that knowledge to tackle some of humanity's biggest challenges.
So we've gone from deciphering the structure of DNA to actually being able to manipulate genes.
Right.
And now even potentially curing diseases. It's mind-boggling how far we've come.
As we wrap up our deep dive into the 8th day of creation, what are some of the key takeaways you'd like listeners to remember?
Well, I think one of the most important is that science is ultimately a human story.
Yeah. Full of passion and collaboration, and sometimes setbacks too, of course. The stories of these scientists,
Right.
they remind us that progress isn't always linear. It's full of twists and turns and sometimes even dead ends. But there are unexpected discoveries along the way too.
It definitely takes dedication and resilience to keep pushing the boundaries of knowledge. It's not just about smarts.
Right.
It's about being curious and persistent and willing to question assumptions.
Exactly. And I think another key takeaway is that these discoveries have profoundly changed our understanding of life itself.
Yeah.
We know now that all living things are connected by this shared language of DNA, this incredible code that shapes everything about us.
It is a unifying concept when you think about it.
It is.
Despite all our differences, we're all part of this grand tapestry of life, woven from those same threads of DNA.
Beautifully put. And now we have this power, right, to read this code and even edit it.
It's both amazing and a bit scary at the same time.
I agree. And Judson, he doesn't shy away from those ethical questions. He challenges us to think about how we use this knowledge responsibly, the potential benefits and risks of genetic engineering and biotechnology.
Absolutely. It's a conversation we all need to be part of.
Definitely.
Well, as we wrap up our deep dive into the 8th day of creation, I encourage everyone listening to keep exploring these questions, read the book, delve into the research,
Yes.
and have those conversations about the future of biotechnology.
Because the story of the 8th day of creation, while it's still being written, we all have a role to play in shaping what comes next.
And that's a great note to end on. Thanks for joining us on this journey through the incredible world of DNA. And until next time, Keep seeking knowledge, keep asking questions, and keep exploring.